Okay, we're picking up from after the exam, continuing chapter 6 on cells, setting up one of the labs that we're going to do. We've been talking about membranes, about barriers, and how they have some control over what goes in and what goes out of them. Now we're going to talk about what makes things move across a barrier. Lots of stuff do where you're not seeing me 3 times a week in class where I can go, this is coming up or that's coming up. You've got to be hitting your own head with this is coming up and that's coming up. So basically, laying in bed, eating breakfast, going from place to place, working on a computer. All she's thinking is, I should be writing. I should be writing. And then it looks like the computer's saying, you should be writing. And she goes, I know. On an atomic level, in any kind of medium where particles are free to move, so in gases or in liquids, there's a tremendous amount of space that the particles move through. Even in a liquid, there's, the liquid molecules are moving around and there's space in between them, so everything's moving, and basically they move until they hit something and then they bounce off and move in another direction. Every once in a while they hit something and they stick to it, and that's part of what makes a chemical reaction happen. But if they hit a medium they can't get through, then they just bounce back. So in a beaker, the glass, the particles of the glass are packed too close together. Anything in the water, both the water and stuff dissolved in the water, are going to hit that glass and bounce back out. Now, if you take a clump of sugar, they used to actually make sugar lumps. You'd actually get them, you'd go to a restaurant, there'd be little sugar lumps. Eventually they wrapped them in paper. For a while they were just, in a dish. It's really tough to find sugar lumps anymore, but you get the basic idea. As a cube of sugar, if you drop it into water, it will start to dissolve. What does that mean? Well, it means that the sugar particles on the outside will start to come free into the water, and when they come free, they just start to move. They move and they hit things and they bounce, and they move and hit things and they bounce. But what that will do is it will gradually spread them out from where they started, where there were a bunch of them, out into the parts of the beaker where there's more space. It moves them from an area of high concentration to an area of low concentration. This is a very low concentration area, and they're not doing it because they know that there's room out there. They're doing that because there's more particles, let me get closer here. There's more particles headed this way to spread out than there are heading back that way to where they started. Now eventually, if you leave them long enough, they're just all moving around inside the beaker. And you couldn't tell where that lump of sugar was to start with, because they're moving in every direction. They will have spread themselves evenly through whatever medium they can spread evenly through. And that is diffusion. Movement of an area from high concentration to an area of low concentration until the concentrations are balanced, if the concentrations can be balanced. Sometimes you can't hit a balance in concentration. Movement of particles from a high concentration area to a low concentration area. Sometimes there's a barrier involved and things are moving through the barrier. If, say, one of the local supermarkets back when it was a really big deal. They opened their store for the first hour to us old people. And so we're in there, we're shopping, we're moving around, and we're, you know, we're gradually moving back out into the parking lot. But then the hour hits that everybody can come in. And up until that point, a bunch of people have accumulated on the outside of the store. And they're ready to go. So when the store says, okay, you guys can come in, there's still old people leaving the store, but there's way more not old people coming in. The high concentration area is moving mostly into the store. The low concentration area is moving gradually out, but the net flow of people is into the store. High concentration area to low concentration area. Because one thing sometimes people get in their head is a high concentration area to a no concentration area. And it doesn't have to be that way. As long as you've got relatively more things over here going that way than are over here going that way, the net movement is going to be that way until you do get a balance. Until eventually, here we're just tracking people. Eventually later in the day, you're getting an even flow of people into the store and out of the store, and into the store and out of the store. And that's not a perfect comparison, but it gives you an idea of how this works. Excuse me. My W's are getting worse and worse. One thing it's really easy to lose sight of is that to start with, there's no water in this cube of sugar. And as the cube of sugar dissolves, as it comes apart, there's a movement of water into where the cube of sugar used to be. Because there's more water out here heading that way than is water in there heading that way. Water also diffuses. It moves from a high concentration area to a low concentration area if it can move in that direction. Remember that cell membranes are water barriers, but they're not great water barriers for most things. Most cells are only active when there's water around them. So they're pretty permeable to water. Bottom line for permeability is you've got some sort of obstruction, some sort of barrier set up. Can this particular thing go through that particular barrier? We got a screen door up, and the wind kicks up a whole bunch of dust, and the dust comes through the screen door and hits you in the face. Because the screen door is permeable to dust, but it's not permeable to mosquitoes. That's why you got it in the door in the first place. If a gust of wind pushes rain into the screen door and you're standing next to it, you get this spray of fine particles because it splits the rain up trying to come through the little holes in the screen. But the screen door is permeable to water. It's obviously permeable to air. Now, air is tricky because we talk about it like it's a single thing. But air is a combination of different kinds of particles, and they all move independent of one another. So the nitrogen in the air, and the oxygen in the air, and the carbon dioxide in the air, and the few other things that are in the air are all moving independent of one another. Everything that's dissolved in the water is moving independent of the other things that are dissolved in the water. Unless there's some reason for them to stick together, which doesn't happen very often, they are all moving, and they're all free to diffuse, but not every barrier is going to be permeable to them. When we do our diffusion lab, we're going to put some plant cells into a situation where we surround them with a salt solution. What is a salt solution? It's water with sodium ions and chloride ions dissolved in it. Because remember, salt can't stay salt in water. It's the two ions split apart and become hydration shell separate. And it turns out that these plants they have a certain amount of chemistry that involves sodium and chloride. They live in fresh water where there's no sodium and chloride around them. So they are not permeable, their membranes are not permeable to salt. Because they don't, if it was, there'd be more salt on the inside headed out than on the outside headed in, and they'd lose all their salt. They're certainly permeable to water. They live in the water. What does it matter? But what we're going to do is we're going to change the concentration of water between the outside and the inside of the cell. When we start, the plants live in fresh water. There's a whole bunch of water on the outside. The concentration says it will flow in, more water going in than going out. The thing is, though, the cells are as full of water as they can get. We'll talk about that in a few minutes. And so the only way for water to come in is to bump water that goes out. But the salt in the salt solution isn't coming in for the same reason that it's not going to let it go out, it's not going to let it come in either. So all you're going to see is a net flow of water, osmosis. Osmosis, moving from an area of high concentration to an area of low concentration. Red blood cells. Red blood cells are a bag full of hemoglobin. Now, all of the cells in your body and your tissue fluid and the plasma in your blood have an equal concentration of water. So the amount of water is always the same. The other stuff is different. But if you're only interested in how the water moves in and out of cells, then everything is in balance. If you take red blood cells and you put them into a beaker of just water, the water will osmose, it'll diffuse into the cells because there's a lot less water in the cells. So for every few molecules that are leaving, a whole bunch of molecules are coming in, and it swells the cells up and pops them. When I first wanted to study how cell membranes worked, this is how they did it. They took a bunch of red blood cells, which don't have a heck of a lot of stuff in them, and they put them in fresh water, popped them. They didn't really need to like pop, 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 pop. They just, because the osmosis popped them. And then they ran them through a filter. All the hemoglobin and the stuff that used to be in the cells goes through the filter. You catch all the membranes, and you got a whole pile of membranes to do, tests on. This is considered a hypotonic solution. It has more fluid in it, more water in it than what's on the other side. Now, typically, your plasma is an isotonic solution. Water can go in and out of these blood cells, but the amount of water in the blood cells is equal to the amount of water in the plasma. The other stuff that's dissolved in the water are different. There's a whole bunch of hemoglobin in your red blood cells. There's no hemoglobin in your plasma, but there's other stuff in your plasma that keeps the water balance equal. So if this is the water level, all this other stuff that's dissolved and all this other stuff that is dissolved is different stuff, but it keeps the water levels equal. So water goes in and out. Now, if you were to take and put your red blood cells in a really salty solution, I talked about doing that in the lab, then there'd be more water headed out than is headed in, because you've dropped the concentration of water around the cells, and they would they'd crumble up. They wouldn't crunch, because they're not crunchy, but they would shrivel. And that's a hypertonic solution. I may ask you that on a bonus question. I'm not going to expect you to remember those terms. To me, the terms are backwards. To me, hypertonic looks like it should mean more water, and it means less water. Our ancestors evolved in the oceans. And so as cells evolved, just because this is, well, let me tell you what evolved and I'll tell you why. The water levels inside the cells match the water levels in the ocean water around them. Again, the stuff dissolved in the water was different, but the water levels were the same. You know, way more salt on the outside than inside the cells. Way more cell stuff on the inside than outside in the ocean water. Why? Well, because if there was an imbalance, if the ocean water was more watery, then there'd always be a net flow of water into the cell, and the cell would have to do something about that. If the ocean water was less watery than what was in the cell, there'd always be a net flow of water out of the cells, and the cells would have to do something about that to sustain. If this is the level the cells needed to live, and this was what they were in the ocean water, then to stay alive, they'd have to deal with the fact that water was going to be leaving. Eventually, things that evolved into the oceans started to move onto the continents, and they did that by working their way up the rivers. Rain outflow, fresh water, much more watery than the oceans. Now, they could have done this gradually. We'll talk about this much later, where coming into tidal areas, coming into estuaries where the fresh water and the salt water mix, they could have come up gradually with ways to deal with more watery environments than having immediately to go into fresh water and deal with. Fresh water is pretty much 100% water. But that is a problem. If you're a living cell in fresh water, then there's going to be more water coming into you than is leaving. That's why if you sit in a bathtub too long, you get all pruny. Is what happens is I mean, you're not that permeable to outside water, but some of it gets in, and it puffs up your skin, mostly in tissue water. And you can actually see the little strands where your connective tissue holds your skin down as you're and your blood is trying like crazy to get rid of the water. Move it away and flush it out through your kidneys. And your kidneys are dumping albumin in to try to, as a thing to dissolve in the water to keep everything even. But back to fresh water. As living things moved into fresh water, they had to deal with the fact that there were always going to be a higher concentration of water around them than inside them. And there's three distinct ways to deal with that. Plants, like the ones we're going to use in the lab. After our plant cells do a little song and dance with the salt solution, where the water tends to leave the plant and shrivel it up inside the cell wall, we'll replace that with fresh water again, and the plants will re-expand. But they won't re-expand to the point where they pop. They will expand until they hit the cell wall. And the osmosis of water into the cell isn't strong enough to break the cell wall, to pop the casing. It's like me giving you a balloon and saying, blow it up, but before you blow it up, stick the balloon into this soda bottle and blow it up. And you'd blow it up until the balloon hit the soda bottle, but unless you got incredibly powerful lungs, you're not breaking the soda bottle, so the balloon is never gonna get more inflated than the soda bottle. And that happens with plant cells. Water does come in, and it does inflate the cells, but they hit the cell wall, and then the only way for water to come in is if water leaves, right? There's, if the store is at full capacity, you can't put people in enough to break the walls. So the only way for people to go into the store is wait for people to leave the store. It's the osmosis of restaurants, I guess. That's one way. And for plants and for funguses who naturally have cell walls, moving into fresh water was not that big a challenge. And for a lot of protozoans that have casings of different materials, it was okay. For other things that don't have casings, then they're like out in the lake in a leaky rowboat. The water's always coming in, water's always coming in, always coming in, and if you want to sustain yourself, you better bail. And so a lot of freshwater single-cell organisms have what are called contractile vacuoles. We'll talk about this when we talk about cell things. And they literally pump water back out. Water's always coming in by osmosis, they're always pumping it back out. So they're maintaining their water levels all the time. Got to work at it all the time. Now with multi-celled systems, they had a different option. This is what happened with our fishy ancestors. As our fishy ancestors moved into freshwater, they mostly coated their outsides with an impermeable to water barrier. They basically waterproofed themselves. I know it's a weird thing to think about, but fish are mostly waterproof. Is that their scales and their skin are waterproof. They don't need to worry about water flooding in through all of their external stuff. Now you can't waterproof everything. You gotta, any place where you need oxygen and carbon dioxide to move through a barrier, you can't block water too. So the gills were not waterproof. The digestive system, again, moving stuff in and out, is not waterproof. So fish do a combination of being waterproof and pumping extra water out with their kidneys, which is what we do too, right? You drink a bunch of water, two things happens. So a whole bunch of water comes into your blood and makes your blood a little bit more dilute. Your kidneys, in order to make this balanced, they put albumin, a tiny little protein, into your blood that brings the other stuff of the water. Let's see. They put stuff in here, and this is your tissue fluid level. This is your dilute blood. Your kidneys are putting stuff into this blood plasma, and it's making the water levels comparatively, right? It's all relative. Drop down to your tissue fluid levels. That you don't really need to understand. I know that's kind of confusing, but it is a way. We always stay in balance. You can actually kill yourself by drinking too much water. You can overwhelm the system, and then water floods into your blood cells and your brain, or into your brain cells, and can kill you. So these are the three things that freshwater organisms had done to kind of resist this. As plants got up onto the land and needed support structures in order to be up where the light could hit them, they used a little bit of osmosis to give them some reinforcement. They actually do blow the cells up like balloons and give them some extra strength. We'll talk about that later. But also, photosynthesis requires water, right? Water and carbon dioxide, they produce glucose. Producer, bottom of the food chain. Well, the carbon dioxide's in the air, but the water, once the plants were on the land, the water was down underneath them, on and in the ground. They needed to get it up to where photosynthesis was happening. Well, rainwater and groundwater is fresh water. It's pretty pure water. So it is naturally going to move into a living organism. We just talked about that. And so that's what it does with the roots and the support structures of a plant. It moves in. Now up here, the plant's using water. Photosynthesis. It's the water's essentially being converted into glucose. So that drops the concentration of water high in the plant. And so the concentration up here is lower than the concentration down here, and it gradually moves the water up the plant. It pushes the water up the plant. That's where root pressure is. The problem is that if I had a huge jug of water and I was lifting it, it gets harder and harder to get farther and farther over my head because gravity is pulling on the water. It does in the plants too. So root pressure is really good to get maybe 3-4 feet off the ground. And then just the gravity of the water, the weight of the water makes it harder to get any higher in a plant. And to get higher in plants, there's all sorts of cohesion, adhesion, literally suction of in tubes and stuff that plants do. But for early plants, they could get pretty decent ground cover just using root pressure of the groundwater osmosing, diffusing into the plants. Okay, we're going to stop it there.